Hello everyone, my name is Mandhri Abeysooriya, and today I'm going to talk about our work in Excel spreadsheet problems in genomic research. So why data reproducibility matters? So in reference to scientific validity, if a result of a study cannot be reproduced, it raises questions about the accuracy as well as the reliability of the findings. When it comes to transparency and accountability, if others can reproduce a study's finding, it increases the confidence and the accuracy of the validity of the study. Building on prior research enables researchers with verifying the findings of the earlier studies, which they can use as a foundation for new investigation. Scientific progress also provides a foundation for building on previous work and advancing of the science. Data reproducibility also improves decision-making, which allow policymakers and industry professionals to make evidence-based decisions that are more likely to be accurate and reliable. Reproducible research is generally more cost-effective, and it can be effective, and it can actually reduce the repeat experiments that has been already conducted. As you can see, maintaining data reproducibility is crucial for advancing scientific research, but it has been challenging when working with complex data structures such as genomic data and when used with certain tools like Excel spreadsheets. One thing everyone forgets is that Excel is not built to analyze big data. One of the key recent Excel disasters in recent years was how Excel caused 16,000 test results of AstraZeneca to be disappeared from spreadsheet due to its limited capacity. Genomic data can be extremely large and complex and often consist of billions of data points, which can be easily exceeding the capacity of a spreadsheet. While spreadsheets offer some formatting options, they may be not sufficient for genomic data. For example, genomic data often contains long sequence of letters, which represent DNA bases, and that can be very difficult for a spreadsheet to interpret. Genomic analysis often involves in complex calculation and statistical analysis that may be beyond the capabilities of a spreadsheet. When it comes to data integrity issues in Excel, spreadsheets are prone for data entry errors, which can have significant consequences in genomic analysis. For example, a single incorrect base pair can lead to inaccurate genetic interpretation. As you could see, using spreadsheets, especially Excel, is not at all ideal to analyze big data in the genomics. So during the rest of my presentation, I'll be talking about how Excel spreadsheet causes gene name errors. When it comes to gene name errors, this is an issue discovered 19 years ago by Zeeberg and his team in 2004. They discovered that the gene names tend to convert to dates, and moreover, recon clone identifier also tend to change to floating numbers. The recon clone identifier is like a unique identifier given to a specific coding sequence of the DNA. After 12 years from this discovery, Seaman and his team in 2016 did an extensive search where they discovered 19.6 gene name errors from 3,597 articles, which has been published from 2005 to 2015. In 2020, HGNZ or Hugo Gene Nomenclature Committee were mainly responsible in assigning the standardized names for human genes, have changed gene names for some genes, which include SEPT1 being changed to SEPTEEN1 and MARCH1 being changed to MARCHF1. Recently, Seaman and his team did even more extensive search where they discovered something new, and that was 5-digit number conversion. Moreover, they also discovered 30.9% gene name errors from 11,117 articles, which has been published from 2014 to 2020. Gene name errors discovered in 2021 is based on the research I conducted for my master's thesis, which was supervised by Mark Seaman. And so this presentation is actually based on the method and results of my master's thesis. So this is an output of an Excel spreadsheet for a genomic paper, which has been published in 2014. The inside the red box are some of the gene symbols that has been affected with gene name errors. And inside the blue box are possibly the correct gene symbols for those affected genes. So even though we were able to assume the correct gene names for some, if you could look towards the end of both red and blue boxes, you can see that it's impossible to assume the correct gene name errors for all the erroneous conversions. So moving on for the method. So this is the framework of our method, where we started by observing the behavior of the spreadsheet software, and then screening the gene name errors that has been converted to dates. And then we did a PubMed central search to find the articles. And then we had software which we used to scan the gene name errors. And then finally a data verifying and visualization. So as the first step, we wanted to observe the behavior of different spreadsheet softwares. So we used the same string of the data in 4 different softwares, such as Microsoft Excel, Google Spreadsheet, LibreOffice, and Genomic. So if you could draw your attention to the March 3 in this blue box, you could see that the March 3 gene tend to convert to a date in both Microsoft Excel as well as on Google Sheets. As for LibreOffice and Genomic, for the same string of the data and for the same March 3 gene, you could see that the gene does not alter in either of these softwares. So therefore, it can be concluded that the Excel and Google Sheets are prone for erroneous gene conversions. So as for the second step, we also wanted to see how many gene name errors are occurring in all eukaryotic genes that are available in Ensemble. Therefore, we downloaded all the eukaryotic gene annotation files from the Ensemble, then extract the gene names and taxa related to each of the species. Then we import the files to Excel and sort the gene column. So by doing so, we were able to find the erroneous conversion on the top, and then we calculated number of erroneous conversion for each taxa and species. As for the third step, we extract the PubMed central articles. In order to extract the PubMed central articles for our study, we screen the PubMed with the keyword genome in titles and abstract. So by adopting this method, we were actually able to reduce the quantity of publication to be screened for gene name errors around 95%. So this is a step where we use the software to scan the gene name errors. So supplementary files from the PMC articles with the extension XLS or XLSX was extracted. As you know, these are the file extension for Excel spreadsheets. And then each of the downloaded Excel supplementary files was opened in R. So all the columns in every sheet was selected and top 20 cells was chosen. For each column, top 20 cells, then we intersect it with the gene name list that we have in our system. And then from that, we were able to identify the species name. And if the number of genes recognized in 20 cells are greater than 4, then regular expression was used to find the erroneous conversion. And lastly, if there is erroneous conversion, then the detail of the paper was logged in a separate script. So this is our final step, which is data verification and visualization, where the suspected erroneous supplementary files was downloaded, and each of these files was opened in R to confirm the error, and the columns which contain the gene symbols was sorted. So verified data was loaded to R for further analysis and visualization. So moving on for the results. This is the output of the result to gene name errors observed across Eukarya and organism. In the table 1, demonstrate the gene name errors across Eukarya for top 5 gene annotation files downloaded from Ensemble. So in here, vertebrate has the highest affected genes and Metazoa having the lowest. In the table 2, demonstrate the gene name errors stratified by organism for top 5 for PubMed central articles that we have downloaded from 2014 to 2020, where the common house mouse has the highest affected articles and East having the lowest. So this is the result from screening the PubMed central for the gene name errors. Figure A shows the articles with supplementary Excel gene list. Figure B shows the article that has been affected to gene name errors. As you could see, there's a similar increment pattern between figure A as well as in figure B. The figure C represents the proportion of the affected articles from 2014 to 2020. As you could see, there's not much of a fluctuation in the proportion of the affected articles, even though this issue has been addressed in 2004 as well as on 2016. So this is the result for the prevalence of the gene name errors across the journals. So in here, I have only selected top 5 journals, which has more than 50 articles with supplementary gene Excel list. As you can see, high impact journals such as Nature Communication tend to contain more erroneous gene names. We also observe some novel type of errors in our study. For example, some genes tend to connect to local language settings, such as a AGO2 gene, tend to convert to August 2, and we believe it may be because ago is August in Spanish. May gene also tend to convert to May 1st, and this may be because May is referred May in Dutch. And TAM41 gene also changed to Jan 41, and we believe it may be because Tamiku is January in Finnish. So there are some other dates that are very unrelated to Excel date genes, such as February 1997, August 97. So by observing the other columns in the Excel spreadsheet on related articles for these errors, we are seeing that they are actually coming from genes names such as set, mark, and dec. So one of the significant findings in our study was the 5-digit number conversion. So some genes, as we know already, that tends to change dates. And upon saving and importing these Excel date genes into a new Excel spreadsheet, 5-digit number conversion could be observed. And I will demonstrate an example in my next slide. So this is an example of the 5-digit number conversion. As you could see in the figure A, so figure 1, the output of the Excel, this is an output of the Excel when stored the genes. Figure 2 is illustrating an instance where the previously stored gene name Excel file getting imported into new Excel spreadsheet. Figure 3 shows how certain genes that has been converted dates to previously now has been changed to 5-digit numbers. As you can see, when gene names change to 5-digit numbers like this, you will not be able to recognize the original gene name, and thereby this affects the data reproducibility. So we have some recommendations to avoid the gene name error conversion. So using scripted analysis where R or Python is always preferred over spreadsheet, since these languages are built to analyze and process big data. LibreOffice is always recommended and is also freely available. And instead of using spreadsheet, you can share your genomic data in flat text files such as CSV, TSV, or TXT. If using Excel is unavoidable, then please do take care in importing data from flat text files. So that being said, you can sort the columns of each data before importing. For example, columns that contains gene names should be formatted as free text and gene expressions as numeric. As you can see, Excel affects data reproducibility, and thereby it's a high time to stop using Excel spreadsheet for genomic analysis. Even though agency has taken initiation to change most error-prone gene names, this will not solve the problem entirely, because there's going to be still a large number of genes that could convert to errors if the user compute is set to non-English language. So while gene name error may seem like a small issue in grand scheme of scientific data analysis, they are an indicator of a larger problem in scientific data reproducibility. Therefore, researchers should consider using specialized software designed to analyze big data to reduce the risk of introducing errors into their research. So that being said, please don't be like Joy. So if you want to know more about our work, how Excel alters the gene name errors, please follow our work on conversation and post-computation biology. Finally, I would like to thank my 2 teams from Deakin for their endless support, and also for Kansas, Ontario, and Data Science Institute of University of Toronto for giving me the opportunity to present our work. Thank you all for listening.